everybody. So you want me to talk about genetics today? Alright. Genetics is about why we resemble our parents, our grandparents, and sometimes even remote uncles and aunts, and yet look very different from them. This phenomenon was observed in plants, animals, worms, insects, and so on, and people were naturally interested in knowing what was happening, and there were theories like inheritance was passed on via blood, or inheritance was passed on as a miniature adult by the male parent, and female parent provided an environment for it to grow. But most prevailing theory was the contributions from both parents were mixed or blended together to produce an individual with intermediate features. With such background, an unknown monk from Austria, Gregor Mendel, was keenly interested in this and was, as a trained botanist, was interested in looking at plants for finding an answer. He rightly reasoned that any plant that he would use should have a few properties, suitable properties, such as it should produce various characteristics with contrasting features. It should be possible to produce the same characteristic generation after generation. It should also be possible that if it was self-pollinating, to bring about cross-pollination. Looking around, he settled down for ordinary common garden pea plant because it showed at least 7 characteristics with contrasting features like round seeds, wrinkled seeds, yellow seeds, green seeds. Thus perfecting his techniques, he decided to start his experiments. His church allowed him to grow these plants over acres of land. He cultivated them in very systematic manner and was all set to start his experiments. Let's start with what he did to round and wrinkled plants. Before any self-pollination occurred, he opened the petals of a wrinkled flower, gently removed all the pollen from that, and introduced pollen from round seeds into the pistil of these depollinated flowers. Close the petals and let it grow. And when these grew and produced pods and seeds, he used these seeds for cultivating F1 generation. F1 generation 2 grew up, produced flowers, and when the seeds turned up, they all turned out to be round in shape. What happened to the wrinkled seeds? He rightly guessed they could not be lost, so he didn't stop here. He allowed this plant to self-pollinate itself. And when these seeds were ready, he used them to cultivate F2 generation. And when F2 generation was ready and seeds came out, there were wrinkled seeds as well, in addition to the round ones, in a ratio of round to wrinkled as 3:1. What happened to the wrinkled variety in F1 generation? He rightly reasoned that for any character to appear, the plant would need both features, 2 features. One of them always dominating the other. In this case, round dominated over the wrinkled variety. So he called round as the dominant feature, giving it a capital P R as the symbol, and wrinkled as recessive feature with a lowercase r for its symbol. Let me introduce you to a few terms here. Round by round and wrinkled by wrinkled are homozygotes, whereas round by wrinkled is known as a heterozygote. Now round by round and round by wrinkled are round in shape, whereas wrinkled by wrinkled only, are wrinkled in shape. In the parental cross, he introduces round pollen into the pistil of a wrinkled flower, thus mixing these two factors. The plant grows as such, and when it is time for it to produce gametes, these two factors, round and wrinkled, separate out or segregate cleanly. That is, there is no mixing, no blending, no intermediate variety at all. Dominant remains dominant, recessive remains recessive. And when he allows them to self-pollinate in the F1 generation, they once again combine, producing a round wrinkled plant. When this plant is ready to produce gametes, both pollen and eggs are of round and wrinkled variety in almost equal number, as shown in the chart. These together produce round and wrinkled seeds in a 3:1 ratio. This is known as Mendel's first law of segregation. Talking of humans, brown eye color is dominant over blue eyes. In a general population, you predominantly see brown-eyed people. Who is a homozygous and who is a heterozygous here, you cannot tell easily by just looking at them. Relatively, blue eyes are rare. They can remain masked over generations until two heterozygotes marry and have children. 3:1 ratio can be misleading. It does not mean one of the 4 children will have blue eyes. All it means is every child to be born here has a 75% chance of being brown and only 25% chance of being blue-eyed. So, Mendel's first law has explained to us what dominant and recessive factors are. It has also explained to us that dominant and recessive factors never mix, never blend to produce any intermediate variety. Now let us look at his second experiment. This is a dihybrid cross where he uses 2 characters in the same plant, round yellow and wrinkled and green seeds. Starting with these true bred plants, procedure is same as the monohybrid cross. In the F1 generation, there are only round and yellow seeds, both being dominant. But in F2 generation, he sees 4 different types of progeny. He reasons out, for this to happen, there must be 4 different types of gametes as well. And that can happen only when each of the equal number of gametes of each character seeks independently any 2 of the other character. That is to say, round gamete can go with yellow or green, irrespective of what the wrinkled gamete would do. This is known as his law of independent assortment. 2 characters here yielded 4 varieties of progeny. Variations will increase as more and more characters get added. Thus, these 2 laws have explained what was surprising to the general public in those days. These 2 laws were not accepted happily by the then scientific community. He was ridiculed and accused of manipulating data. He toiled hard, producing tens of thousands of pea plants, counting millions of seeds, confirming his data again and again, giving them statistical meaning. In early 20th century, when others confirmed his results working on flies, corn, worms, etc., today he is known as the father of modern genetics. Friends, this is only the beginning of a fascinating science called genetics. I hope you have understood a few basics. Thank you all.